Cell communication. There are two kinds of communication in the world of living cells: intercellular signaling and intracellular signaling. Intercellular signaling is communication that occurs between different cells. Intracellular signaling is communication within the same cell. There are 4 categories of chemical signaling found in multicellular organisms: paracrine signaling, endocrine signaling, autocrine signaling, and direct signaling across gap junctions. In paracrine signaling, a cell targets a nearby cell. In endocrine signaling, the cell targets a distant cell through the bloodstream. In autocrine signaling, a cell targets itself. And in direct signaling across gap junctions, a cell targets a cell connected to it via a gap junction. Signals that act locally between cells that are close together are called paracrine signals. Paracrine signals move by diffusion through the extracellular matrix. They elicit quick responses that last for only a short period of time. These signals are degraded quickly by enzymes or removed by neighboring cells. Examples of paracrine signaling include nitric oxide signaling in blood vessels, synaptic signaling of neurons, local skin allergic reactions, the blood clotting system and tissue repair, and wound healing. Signals from distant cells are called endocrine signals. Endocrine signals originate from endocrine cells. Many endocrine cells are located in endocrine glands. Endocrine glands include the thyroid gland, the hypothalamus, and the pituitary gland. Endocrine signaling is done in the form of hormones. Hormones produce a slower response and have a longer-lasting effect. Hormones can act over long distances and travel through the bloodstream. The ligands released in endocrine signaling are called hormones. Hormones can be thought of as signaling molecules that are produced in one part of the body but affect other body regions some distance away. Autocrine signals are produced by signaling cells that can also bind to the ligand that is released. This means the signaling cell and the target cell can be the same or at least a similar cell. The prefix auto means self, a reminder that the signaling cell sends the signal to itself. Autocrine signaling occurs during early development of an organism. This signaling ensures that cells develop into the correct tissues and take on the proper functions. Autocrine signals regulate pain sensations and inflammatory responses. Autocrine signaling plays a key role in cellular apoptosis. If a cell is infected with a virus, the cell can signal itself to undergo programmed cell death or apoptosis. This kills the virus in the process and preserves the functioning of nearby cells. Gap junctions in animals and plasmodesmata in plants are connections that occur between the membranes of neighboring cells. These are fluid-filled channels that allow small signaling molecules called intracellular mediators to diffuse between the two cells. This allows the cells to respond in a coordinated way. Small molecules or ions such as calcium ions are able to move between the gap junctions. However, larger molecules like proteins and DNA cannot fit through these channels. The specificity of the channels ensures that the cells remain independent but can quickly and easily transmit signals to one another. An example of this coordination is the coordination of the heartbeat in cardiac muscle cells. There are 2 types of receptors: internal receptors and cell surface receptors. Internal receptors are also known as intracellular or cytoplasmic receptors. These are found in the cytoplasm of the cell and respond to hydrophobic ligands that are able to cross the membrane. These ligands bind to the proteins inside the cell and act as regulators of mRNA synthesis or transcription to mediate gene expression. Gene expression is the process of transforming a cell's DNA into a sequence of amino acids, which ultimately forms a protein. When the ligand binds to the internal receptor, the ligand-receptor complex moves into the nucleus. Once in the nucleus, it binds to a specific regulatory region of the DNA and promotes gene expression. Hydrophobic signaling molecules typically diffuse across the plasma membrane and interact with intracellular receptors located in the cytoplasm. Many of these intracellular receptors are transcription factors that interact with DNA in the nucleus and regulate gene expression. Cell surface receptors are also known as transmembrane receptors. These exist on the surface of the cell and are partially embedded in the membrane. Cell surface receptors bind external ligand molecules. They perform signaling transduction, which is an extracellular signal that is converted into an intracellular signal. Each cell surface receptor has three main components: an extracellular domain, which is outside the cell, a transmembrane domain, which is a hydrophobic region that spans the membrane itself, and the intracellular domain, which lies inside of the cell. There are three general categories of cell surface receptors: ion channel linked receptors, G-protein linked receptors, and enzyme linked receptors. Ion channel linked receptors bind a ligand and open a channel through the membrane that allows specific ions to pass through. When a ligand binds to the extracellular region of the channel, there is a conformational change or change in shape in the protein structure that allows specific ions to pass through. Gated ion channels are located in the plasma membrane and allow for the controlled flow of ions into and out of the cell. The channel proteins remain closed until a signaling molecule binds to the channel protein. When this occurs, the channel protein changes its conformation, allowing ions to flow into or out of the cell. When the signaling molecule is released, the channel protein resumes its closed conformation, preventing the ion flow. G-protein linked receptors. Once the ligand binds to the receptor, the resulting change in shape activates the G-protein. The G-protein releases guanosine diphosphate or GDP and picks up guanosine 3-phosphate or GTP. The subunits of the G-protein then split into two subunits. One or both of these G-protein fragments may be able to activate other proteins as a result. After a while, the GTP on the active subunit of the G-protein is hydrolyzed to GDP, causing both subunits to become deactivated. The subunits then reassociate to form the inactive G-protein, and the cycle begins once again. Enzyme-linked receptors are cell surface receptors within intracellular domains that are associated with an enzyme. The intracellular domain of the receptor itself is an enzyme. Other enzyme-linked receptors have small intracellular domains that interact directly with an enzyme. When a ligand binds to the extracellular domain, a signal is transferred through the membrane, activating the enzyme. Activation of the enzyme sets off a chain of events within the cell that eventually leads to a response. Signaling molecules are produced by signaling cells. The subsequent binding to receptors in target cells act as chemical signals that travel through the target cells to coordinate responses. The types of molecules that serve as ligands are incredibly varied and range from small proteins to small ions like calcium. Thank you for watching.